Hello, this is Dr. James Camp at Lee College in Baytown, Texas, and this is my lecture on the nonspecific immune system. The human immune response includes three lines of defense: barrier methods, nonspecific immunity, and specific or acquired immunity. Where is that immune response? Well, we've covered already in class that there are 5 kinds of white blood cells circulating through the blood, each of which contributes something to your immune response. But these immune cells don't only exist in the blood. They have their origins, of course, in the red bone marrow in such places as your hips. But then they circulate through the blood, and they also circulate through the lymphatic system, through the lymph. And the lymph nodes are full of certain specific immune system cells, such as lymphocytes, macrophage, and natural killer cells. Lymphocytes, you'll recall, are part of the specific immune response, the immune system response that tracks down and destroys specific pathogens that the body has been exposed to before. In this lecture, we're mostly going to focus on the non-specific immune responses. Other places in the body have their own immune systems, though. There are macrophages that are found in the alveoli and in other places such as the liver. And there are these unusual cells called dendritic cells found in the skin that play a role in the immune system. And then in almost all of your connective tissues, at least all of the fibrous connective tissues, you'll find these cells called mast cells that serve as early warning systems to alert the rest of the immune system that an inflammation needs to take place. All right, so that tells us where the immune system is. What are the pieces of the immune response? The immune system can be broken up into 2 sections based on when and how that immunity is acquired. One portion of the immunity is called innate immunity. Innate has the word "nate" in it, as in neonatal or natal, having to do with birth. So innate is the immunity that we're born with. The opposite of that would be immunity that is acquired over the course of our lives. Because innate immunity is immunity that we're born with, it does not have any ability to respond to things that have happened after you're born. For example, to exposure to one specific pathogen. So this is also known as your non-specific immunity. Acquired immunity, on the other hand, has the ability to adapt to certain specific pathogens, and so this is also known as your specific immunity. It's also sometimes called your adaptive immunity because it changes its response over time depending on what it's exposed to. Those 2 types of immunity can further be divided into 3 lines of defense. Your body's 3 attempts or 3 chances to remove a pathogen or block a pathogen from infecting you. The first line of defense are what we call the barriers. Okay? As with safer sex, barriers are the first thing to try. So the most reliable thing, intact skin and intact mucous membranes form barriers to keep an infectious agent from getting into your body. But beyond that, we also have certain chemical and biological barriers built into our body that we'll talk about in a little bit. The second line of defense involves your granulocytes, macrophages, and certain cells called natural killer cells, NK cells, that seek out and hunt down and destroy cells that look abnormal, either infected with a virus or perhaps cancerous cells. These are able to engage in things like phagocytosis or in inducing cell death, so they can destroy invading or unhealthy cells. They can also cause larger responses like inflammation or fever that bring in the heavy weapons to take care of an infection that's getting out of control. Finally, your third line of defense are your lymphocytes, which include the B cells that produce antibodies and the T cells that produce what we call cell-mediated or cell-directed responses. All right, let's take a little bit closer look at those barrier methods. The epithelial barriers are our first chance to defend against an attack by some sort of pathological agent, and generally they're very effective. Unbroken skin, for example, forms a complete physical barrier, and as long as you don't have any cuts or abrasions on your skin, it's a pretty good way of keeping bacteria out of your body. But what about those parts of your body that aren't covered by skin? The nasal cavity has hairs that protect against large particles getting in, and there's a mucous membrane inside that protects from smaller microbes getting from your nasal cavity into your body. Your eyelids serve as a physical barrier, but they also contain an enzyme called lysozyme that to lyse something is to break it apart, and so it's an enzyme that tries to break apart bacteria. That same enzyme is found in your saliva. Then there are other chemical barriers. Stomach acid and acid in the urinary tract both make for a very uncomfortable environment for most microbes to grow in. And then you'll see that a couple of different places on here, there are some biological barriers. They call them, in this slide, normal microbiota. You've probably heard them called good bacteria or probiotics. These normal microbiota are found all along your nasal cavity. They're found throughout your colon, and they're also found in the vagina as places that, the idea is that if friendly bacteria that do good things for your body live in those places, then they will crowd out the bad bacteria, or even in some cases actively defend your body against the bad bacteria, because like all of us, they want to defend their home. Okay, so that takes care of the first line of defense. What about things that get past it? What if you have a cut in your skin, or a tear in your nasal membrane, or something gets into into the deeper parts of your body? Then you need a second line of defense, and that's where all of these non-specific responses come in. One non-specific response are phagocytes. Remember that neutrophils are phagocytes that will track down and gobble up bacteria. Macrophage are much larger phagocytes that will gobble up things as large as other human cells. The way this works is that the phagocyte finds an infectious agent through a process called chemotaxis. Okay, chemo referring to chemicals, taxis, taxi is to move towards something. So it moves toward a chemical attraction from that molecule. That bad guy is phagocytosed into a little vesicle called a phagosome. The phagosome is merged with a lysosome. Remember from ANP1, lysosomes are parts of cells that contain powerful digestive enzymes that are deadly to the cell. Well, you mix those enzymes with a bacteria, and guess what? A couple of steps down the road, you have digested bacteria. And what portions of the digested bacteria the cell can't use on its own are exocytosed and taken care of as debris, as waste products that the body will get rid of. Another way of looking at this phagocytosis, it's a 5-step process. Chemotaxis occurs, in which the microbe is letting off tiny chemical signals around it, and the neutrophil or the macrophage detect those signals and move toward it. Adherence, the neutrophil sticks to the bacteria. Ingestion, the bacteria is pulled into one of those phagosomes that we talked about on the previous page. Digestion, the phagosome is mixed with enzymes from a lysosome that start to digest the bacteria, and ultimately that leads to killing of the bacteria. Another second line of defense goes not against tiny bacteria cells, but against our own cells that have become sick. If an unhealthy cell, if you have a human cell that has become unhealthy in some way, if it's detected by a natural killer cell, for example, the natural killer puts out some molecules called perforins that perforate, make a hole in the cell membrane, and then other molecules called granzymes that come in through that hole and proceed to kickstart a process called apoptosis, essentially programmed cell death or cell suicide. The cell takes itself apart in a manner that won't do any harm to its host, and what you have left is a little squiggle of cell debris that the body can recycle and reuse to make new cells or do other things. Why would you want to do this? Well, if a bacteria or a virus has taken over one of your cells and turned it into a bacteria or virus factory, then you don't want that cell around. The easiest way to get rid of that virus or bacteria is to kill the cell that it's inhabiting. So going through this process of apoptosis removes an infected cell from the body and in the process kills the pathogen that's inside it. We've talked briefly about eosinophils as being a non-specific line of defense that are anti-parasitic. Eosinophils release toxic chemicals that would try to destroy, for example, a Giardia protozoan or a parasitic worm. They're not entirely good at this. Clearly, if you get infected with a tapeworm, for example, it can stay in your body for quite a long time, but the eosinophils do manage to slow them down a bit. So those are some of our cellular second lines of defense. We also have some biochemical second lines of defense. One of them is a molecule called interferon. When a cell gets infected with a virus, it knows that something wrong is going on. And so it sends out these distress signals called interferons saying, I'm being interfered with. Okay? They cause other normal cells to put up their own antiviral barriers, okay, to do things that will try to protect against the virus getting into that cell or replicating inside that cell. They also attract either your NK cells to come over here and release that apoptosis-inducing attack we talked about on the previous page, or they might induce a macrophage to come over here and gobble the infected cell up and take it apart in that way. Another biomolecular second line of defense is a protein called complement. Complement acts in a whole lot of different ways. It can stick to a pathogen and therefore mark that pathogen for removal by a macrophage or another phagocyte. Complement proteins can also form a perforation into a cell that will actually lyse the bacteria. Boom, boom, no more bacteria. Perhaps the most important function of complement, though, is that when one of your body's antibodies has detected an antigen, the complement will attach to the antibody and then cross-link it to other antibodies, to circulating erythrocytes, to something that will carry that antigen, that pathogen, to, for example, your liver or your spleen, where it can be phagocytosed and removed from the body. All right, and then we have a couple of second lines of defense that are bigger than a single molecule or a single cell. They are whole processes that kind of take over a tissue or a body for a little bit and try to give the good guys an advantage over the bad guys. Inflammation is one in which a basophil or a mast cell is going to release molecules. Okay, the first molecule it releases is called histamine. You've heard of antihistamines that try to prevent basophils from doing this in the context of an allergy situation. But in the context of a damaged tissue or an infection, histamine is a very good thing. It increases the permeability of your blood capillaries so that when the white blood cells reach their destination, they can get out of the blood into your tissue, and you have a cleanup crew to come take care of the problem. The basophil also at the same time releases heparin, which is a molecule it's an anticoagulant molecule. As soon as you open up these permeable capillaries, that's going to look a lot like damaged blood vessels bleeding out into your tissue. So you need to tell the blood not to coagulate in this particular case, and that's what heparin is good at. Heparin, by the way, is also given as a drug in cases where a patient has too much coagulation. Heart attack patients, for example, often get units of heparin. And then finally, the The basophils also release eicosanoids, a subset of which are called prostaglandins. These increase inflammation in the area. There's sort of a positive feedback loop that recruits more basophils, more mast cells, more tissue responses that increase the inflammation. However, they do have a side effect. They cause pain. Eicosanoids set off your pain nerves like nothing else, and you have this problem of tissue pain coming along with the rest of the cleanup operation, which is why a lot of your anti-inflammatory drugs are actually anti-eicosanoids or anti-prostaglandins. There are 4 characteristic signs of inflammation. Those are redness, which is because of the increased blood flow to the area. Pain, which is from those eicosanoids we just talked about. Heat, also from the increased blood flow to the area. Blood is about a degree warmer than your core body temperature, or rather than your peripheral body temperature, so it tends to increase the heat of your tissues. And then there's also swelling. As you open up your permeable blood vessels, more fluid is going to leave the blood into the tissue, and that's going to cause the tissue to swell. There are 4 steps involved in inflammation. Exactly how those steps are arranged varies in my experience from textbook to textbook, but here's one version of them. Step 0, of course, is that an injured tissue situation occurs. In this case, they've drawn a little skateboarder who has skinned his knee. So we've got an open part of the skin with lots of microbes. They're trying to invade the tissue. The first thing that happens is these molecules produced by the inflammatory cells and by the damaged tissue reach the mast cells in the tissue and the basophils circulating in the blood. And they're going to release those inflammatory factors we just talked about. They're going to release histamine and heparin and eicosanoids. Well, what is the histamine and heparin going to do? But it's going to dilate the blood vessels, so it's going to open them up a little bit for more blood cells to get through. It's going to increase the permeability. It's going to open doors here and here and here and here for things to get out of your blood. And it's going to display these cool little things called cell adhesion molecules, little hooks on the surface of the cell that passing white blood cells can use to grab onto and pull themselves out of the tissue. Okay, once they do that, once your neutrophils grab onto these little hooks, they start poking a little bit of their tissue, a little bit of their cytoplasm through that hole that we've created, and eventually the entire cell makes it into the tissue. There are 3 steps in that we call margination, that is moving to the margin of the cell. Diapedesis, that's sticking, literally it means a foot through, sticking a foot through. A little bit of the cytoplasm goes through at a time. And then finally chemotaxis, that cell senses these same molecules that were released by the bacteria and the injured tissue, and it starts moving in that direction. And then once the cell has reached there, it starts phagocytizing the bad bacteria there. Incidentally, when you find pus in a wound, what that is a neutrophil that has gobbled up a number of bacteria until it's kind of gotten overloaded and died. And so the pus is actually white blood cells that have done their job cleaning up your tissue. Finally, we have this situation where plasma proteins start getting delivered to the scene. Remember, plasma proteins include lots of different things. They include antibodies, they include carrier molecules, but they also include clotting factors and fibrinogen. And so over time, one of the jobs that these plasma proteins are going to do is to form a clotting barrier around this this tissue, this damaged tissue area, which you would think of as a scab, that will lock these bacteria out of the wound so that they don't get further into the body. However, some of them will, and so the other thing that's delivered out here at this time is fluid called exudate. Okay, exudate is extra tissue fluid or extra plasma that becomes extra tissue fluid that mixes with all of these little bacteria that shouldn't be there and washes all of that out into the lymph, where it can go to the lymph nodes and be taken care of by all the macrophages and immune cells there. That's where it will also first encounter the specific immune system and hopefully stimulate a full-blown immune response if this infection starts to get out of hand. Okay, we've talked about this already. The inflammatory response is annoying in several ways. We've got edema, fluid swelling, we've got heat and redness resulting from blood flow, and we've got pain from all those inflammatory chemicals running around. And that's the reason why a lot of people take anti-inflammatory drugs to try to calm down an inflammation. But it's my opinion that we shouldn't really be so trigger happy with the anti-inflammatories, especially in the case of an actual open wound like the one pictured here. The inflammation response is, after all, there to help you. It's there to do some good. So lay off the Advil for half an hour or so and let that immune response do its job. Okay, and then the last non-specific response to infection is inflammation. I mean, is fever, which is sort of like inflammation writ large. It's a heat and what happens is that basically the hypothalamus, which we know regulates your body temperature, senses these inflammatory chemicals in the bloodstream, and it increases the set point for your body, the temperature set point for your body. So instead of, being at a happy, 96 degrees where you normally would be, now maybe you're up at 101 degrees. Okay. So instead of being at 98, you're now up at say 101. Why would your body do that? It's uncomfortable to have a fever. Obviously, it's not your body's normal operating temperature, but fever has a number of positive effects on your body. It inhibits microbe reproduction. Bacteria don't like being at 101 degrees either, and so they slow down. But even more importantly, it enhances your immune response. Many of your immune cells actually operate faster and better at 100 degrees than they do at 98 degrees. And it accelerates some of the processes that cause tissue repair. So it can help you close those wounds a little bit faster and start getting the bad guys out of the way. All right. So that concludes our lesson on all of the second lines of defense, all of the the first and second lines of defense, all of the non-specific ways that your body has for fighting infection. Come to class on Tuesday, and or come to class next time, and we will talk more about the specific ways in which your immune system defends your body. Thank you very much, and have a good day.